The first part of protein synthesis is something called transcription. The way I try to remember this, or my learning tool, is I think of prescription. A prescription is something that a physician will write in order to get a medication from a pharmacy. So transcription rhymes with prescription. It's not the actual medication, but it's the code for the medication. They describe how much medication you need, when to take it, and how to take it. So let's get a diagram of the big picture. We have the DNA in the nucleus. It's converted to messenger RNA, which is then converted to an amino acid, which is then converted into a protein. And we'll remember that a protein is basically a chemical message. We are living chemistry, and our body operates through chemical messages. DNA to messenger RNA takes place in the nucleus, whereas amino acid to protein takes place in the cytoplasm of a cell. This half is called transcription. We are writing the code. And just like your foreign language class, this half is translating, or called translation. Just like you do in foreign language, you convert from one language to another. Let's review some vocabulary. A codon is a set of 3 nucleotides. Our nitrogen bases in a nucleotide are adenine, thymine, cytosine, and guanine. Basically, one codon is converted into an amino acid. There's our vocabulary there. And then amino acids linked together to make a protein. So basically, a gene codes for a specific protein. And again, proteins are our chemical messages. They may include things like hormones, body chemistry, directions on how to grow and develop, etc. Let's take a look and see how DNA codes for messenger RNA. So for example, our 4 nucleotides or nitrogen bases are C, G, A, and T. When we convert this to messenger RNA, C converts to G, G converts to C, which we're familiar with, A converts to U, and T converts to A. So we have a new nitrogen base that we haven't seen before, and this is called uracil. There is no thymine in messenger RNA. So let's just go over what messenger RNA stands for. Little m stands for the messenger. Instead of deoxyribose as the sugar, this has what's called ribose sugar. It is single-stranded. It contains uracil. And it can leave the nucleus through the nuclear pore. It can move around. Remember, DNA is so precious, it never leaves the nucleus. Here is a strand of DNA. You can see that it reads GAGCAT. So GAGCAT is our message. It's been opened up so that the messenger RNA can come in and make its strand. Again, remember, messenger RNA is single-stranded. So in this case, it's going to be reading, it's going to come in one at a time, it's going to join in here G, U, A. So this is that strand of messenger RNA, okay? This is just going to be a very short, this is called a codon here. GUA is our strand. This will then leave the nucleus and continue on with protein synthesis. But we want to learn what GUA codes for. This is called a codon chart. It's comprised of 3 different areas that we read from. If you take a moment, just take a quick peek in here, and you'll notice that is not symmetrical or identical. There's words in here such as stop, and then there's all these 3-letter abbreviations. These are all the abbreviations for the amino acids. The stop means the end of a message because, again, this is a code, and we need to know when to start and when to end a chemical message. Each of these stands for the 20 amino acids. And again, we don't have to memorize this. This is a tool that scientists use. So LEU stands for leucine. So we tend to use the words LEU. So going back to the codon chart here, we need to learn how to use this. What I recommend doing is putting a star, and this is for messenger RNA only. You cannot use DNA. It has to be converted into messenger RNA. So in our last video, we had CAT was our DNA, and GUA is our converted messenger RNA. So GUA, this is the first base, this is the second base, and this is the third base. The way you use a codon chart is we're going to look at the first base. So we're going to see up here it says first base. We're going to find G. Here's G, so we know it's somewhere in this row. The second base is U. So I go up to second base here, U. I know it's somewhere in this box. And the last base is A. So I'm going to come over to the far side and I find A, and I bring it over. And the amino acid is VAL, which is valine. So just to see if you can get this, so what if I did UAA? Take a moment and see if you can figure out what it is. Pause the video if you wish. But U, it'd be somewhere in here. UA, so it's somewhere in here. A. UAA, and that stop.